So let's say that we have a group of offspring and we want to determine what the recombinant phenotypes are. So we can do this based on our knowledge of what happens in those chromosomes. So remember, some of those chromosomes exchange information. So 2 of our phenotype combinations are going to be parental and 2 are going to be recombinants. Now remember also that recombinants occur less frequently than parentals do. Now this is because only 2 of our tetrad, or a group of recombinant chromosomes, is actually involved in the crossing over. So we can never have more than 50% recombinants because only 50% of the chromosomes are actually recombinant. And on top of that, we're always going to be less frequent than 50% because not all recombinations are going to occur between the 2 genes that we're talking about. And sometimes we might not see them just by chance, or we might miss a few just by chance. So we can always determine the recombinant phenotypes by the less frequent occurring combinations. So on the top here, we've got just some random combinations: gray body and red eyes. One of these is going to be one parental combination. The other, yellow body and white eyes, is going to be the other. Now, we don't even necessarily need to know what are the mutants here because all we want to know is which ones are the recombinants. If we look at these other combinations, gray body with white eyes, we have a recombinant of 2 of those parental phenotypes. Yellow body and red eyes is the other recombinant because this occurs less frequently than the parentals. Also, when you are close together on a chromosome, there's less chance of a crossing over. So 2 traits that are close together are less likely to make recombinants. And again, this is related to there being a smaller space between them within which a crossover can occur. On this one on the right, we have a closer number of recombinants to parentals, but you can still tell which one is less. We have less recombinants than we do parentals. So these 2, red eyes and long wings and white eyes with miniature wings, are going to be our parental, and the other 2 combinations are going to be our recombinants. So to summarize that last slide, we only get 2 recombinant chromosomes. So 2 out of the 4 chromosomes are going to be recombinant, but recombinants are never more than 50% of the offspring, and that's because only 50% of our chromosomes are recombinant. Again, recombinants are also less because crossovers may occur outside of the genes. And anytime you have a crossover on the outside of the gene, you're just going to get the parental gametes. You don't get any recombinants. This slide actually probably was better before the previous one, but that's okay. All right, so we can use the number of recombinants in order to calculate a relative distance between genes. Now, we do this starting with the steps that we just did on the slide before. The first thing we need to do is determine the parental phenotypes and to determine the 2 recombinant phenotypes. Then we're going to calculate the frequency of recombinants. Frequency is really just a percentage as a decimal. So we are going to divide the number of recombinant offspring by the total number of offspring. That's going to give us a frequency. Frequencies are always between 0 and 1. And then if we convert it into a percentage by multiplying it by 100, we are going to get a map distance. Now we can call these map units. They might also be called, or that would be MU, or sometimes these are called centimorgans. Morgan for Thomas Hunt Morgan, who discovered the white eye mutation in sex-linked traits. So this is written as cm, centimorgan. So because this is a distance, we have to give it some units. So just like you would say miles, kilometers, inches, whatever, we need to give it a unit. So we are going to divide number of offspring by total number of offspring. So let's look at our example from the previous slide. So for the one on the left, our recombinant frequency is going to be equal to our number of recombinants divided by our total. So the recombination frequency for this one on the left is going to be 17 plus 12, because remember our least frequent are the recombinants, divided by the total number of offspring, which is 2205, and then we're going to multiply that by 100. So this works out to 29 over 2205 times 100, which is equal to 1.3 map units. So this is a relative distance. We'll talk about absolute distance maps in a little bit. This is a relative distance. So these ones are actually very close together. They're only approximately 1 unit apart. So on the right here, our recombination frequency. First, we determine our recombinants. So that's these guys here, the smaller number of offspring with those traits. So recombination frequency is going to be equal to our our number of recombinants divided by the total. Then we're going to multiply that by 100. So 401 plus 318 divided by 2205 is going to be equal to 719 divided by 2205 times 100, which is going to be 32.6 map units. So you can see these are farther apart than over here where they are 1.3 map units. And you get more recombinants because they're farther apart. There's a higher likelihood that recombination is going to occur between those genes. So let's do one more example. This is from a different textbook. So when you have 2 genes, you can use the recombinants to give you a relative distance between those genes. So we have some chromosomes here, we have some traits, we have long bristles and gray bodies, so these are like little hairs on the fly, and then we have the mutant or short bristles and ebony body. So they are located on the same chromosome, and we can determine how far apart they are by calculating the recombinant frequencies. Now, when you only have 2 genes, it doesn't really give you much information. All you can really figure out is whether or not they are in fact linked and how far apart they are. This diagram here is really just sort of arbitrary from our perspective, because having only 2 genes only tells us how far apart they are from one another. It gives us no relative information to other genes. But we can still figure out how far the bristle and body color genes are for these flies. So our relative distance is going to be our recombination frequency. So our number of recombinants is 76 and 75. So recombination frequency equals 76 plus 75 divided by our total number of offspring, which is 537 plus 542 plus 76 plus 75. So this is equal to 151 divided by 1230, and remember we're going to multiply by 100, which is going to give us a recombination frequency of 12.3, and this tells us that these are located 12.3 map units apart on this particular chromosome. So let's continue building our genetic map by looking at 3 genes. Now this is called a 2-factor cross, and in a 2-factor cross we are going to look at the relative locations of 3 different genes. In this case we are looking at body color, eye color, and wing shape. So these are some new mutants. We've got some different ones here. We've got black-bodied again. We've got purple eye and red eye. And then we've got vestigial wing, which is similar to the apertius wing. It's really just a much smaller wing. And then, of course, we've got our normal body and wing color. So to do this, you calculate, or to use this information to make a genetic map, you calculate the recombination frequency of the 2 different genes. Or sorry, the 3 different genes. So we're going to calculate the recombination frequency between body color and eye color, and then we're going to calculate the recombination frequency between wing shape and eye color. And this is going to allow us to determine in which order they occur and how far apart. Now we can actually do this for both of the traits. Our book example only does it for one of the traits, and they tell us that the wing and purple eye color are located 12 map units apart. So they're telling us that bit of information, but they have calculated it the same way. Just for simplicity's sake, they're only showing us one calculation. So we already know that vestigial wing and purple eye occur 12.1 map units apart. So let's figure out how far apart the purple eye and the black body are. And we do this with recombination frequency. So again, we determine our recombinants, the least frequent, divide that by the total number of offspring, multiply that by 100. We get 6.6%, which tells us that they are 6.6 map units apart. So let's go ahead and work this out. So recombination frequency between black bodies and red eyes is going to be 35 plus 31 divided by 1,000 times 100 is equal to 6.6%, or 6.6 map units as I have been writing it, but it's a frequency. So then we were given the frequency between the vestigial wings and the purple eye. This should be a P right here, sorry. Vestigial wings and the purple eye, which was 12.1%. Now, we also need to figure out the distance between the vestigial and black wing. So if we go back to our map over here, we have they've given us the distance between vestigial and purple, and we've calculated the distance between purple and black. Now, if we are going to order these genes, we also need to know the distance between the third combination. In this case, the distance between vestigial and black. So just like they gave us the distance between vestigial and purple within our text, they also give us the distance between vestigial and black. They tell us that that is 18.7 map units. So they would use a chart just like this one here that gives the number of recombinants. You would calculate it and calculate 18.7. Now, to determine the order on the map, it's basically a logic problem. So the largest distance is 18.7. That means that the vestigial gene and the black gene are the farthest apart. That means that purple, the purple eye color must be in the middle. Now purple and black are only 6.6 apart. So that means that purple has to be closer to black. So really this last step is just a logic problem. You have to look at it and determine which ones are the farthest apart and the relative position of the one that's in the middle. I am going to ask you to draw it in relative position. We have roughly 6, 12, 18, which means that The purple is about a third of the way of the total from purple. I'm sorry, from black. So I also want you to label the distances. So vestigial and purple are 12.1 map units apart. Purple and black are 6.6 map units apart. And the whole distance is going to be the 18.7 map units apart. So just to make this all nice and concise, step 5 is just to reason out the map. So if we go back, this slide here gives all of the steps for the 2-point test cross. So we need to know the recombination frequencies between all 3 of them, and then we logic through where they are located. Now, you are most likely going to be given all 3 sets of phenotype information. Each one is just determining the recombination frequency, and we've done many, many examples of that now. So each one is determining the recombination frequency. Then you logic out the map, you reason out the map. All right, so this genetic map is a relative map. We know that vestigial is closer to purple than it is to black. We know that purple is closer to black than it is to vestigial. When we do a 2-factor cross, 2-factor crosses often underestimate the distances. And so we can take it one step further and do a 3-factor cross in order to get a better estimate of our map distances. Now, the reason that they underestimate the distance is that we can actually have double crossovers occur along a chromosome. So this chromosome shows A, B, and C, 3 genes with the heterozygous homologous chromosomes A, B, and C. And when there is a recombination in between, we get these chromosomes flipped. So we now have big A with little b, little c, and little a with big B, big C. So we've got our parentals. These ones are not involved in the crossover, and then we've got our recombinants. But sometimes you can actually get a second crossover event occurring at the same time between B and C. Now this is because recombination or crossovers in meiosis actually occur at multiple locations along the chromosome. So when we went through chapter 6, we really sort of just assumed that this crossover was only happening like once on each arm, and that's how we drew it. And really we do that for simplicity's sake. But actually, depending on the length of the chromosome, you can have many crossover events, and that is very tightly controlled within the cell, how many crossover events you're going to have and where they're going to happen. But we can actually have a double crossover event, which means there are more recombinants than we are seeing. Now, the reason for this is that it actually can flip back some of the genes. And so you actually get A back with big C, even though you flipped A with little c initially. So you can't see all of the recombinants that are occurring. You actually don't see the big A, little c recombinant because it flipped back. So we have to ask, was there no crossover? Was there just a single crossover? Or were there 2 crossovers and the big A, big C combination came back? And this is going to give us 2 pieces of information. Knowing whether or not there were double crossovers is going to give us a better map distance. And it will also tell us which gene is in the middle. So I'm going to do this cross with the double crossovers. This is called a 3-point cross in a separate video. So I'm going to skip over it for now. And you can either pause the video and go watch it now, or you can finish out this video. It's not too much longer. And then go watch it in a little bit. If you don't watch it now, it's going to be just fine. So the presence of one crossover actually reduces the likelihood of a second crossover. This is called interference. So interference actually reduces the expected number of double crossover events. Because when you have one crossover, it sort of limits how close you can have another crossover. So we are actually not going, you can actually calculate the level of interference in something called the coefficient of coincidence. We are not going to do that right now. I just want you guys to understand that when you have a crossover event, it can interfere with producing a double crossover. Now, they're not really sure why this happens. It probably has something to do with controlling how frequently crossovers occur so that you don't get too many crossovers. But the actual mechanism for how interference works is not really understood. So we have been talking about relative maps on eukaryotic chromosomes. We can also make genetic maps of bacteria. Now they work a little bit differently because bacterial chromosomes are circular, whereas eukaryotic chromosomes are linear. So the genetic map for a bacterium is actually going to be circular, and there's not necessarily a start and a stop. You could put a start really at any point along this chromosome. And it depends on where you actually start making your map. So this prokaryotic map is E. coli K12. This is a commonly used E. coli in the lab. And the way that you make a map of E. coli is by allowing one E. coli to infect another with its circular plasmid. Now we will talk about how this works in chapter 11, so in our next unit. So we're not going to go into great detail now about how or why this happens, but you can make a map by allowing this bacteria on the left to infect the cell on the right. So what happens is this circular chromosome gets a little cut in it, and it is released from the cell and put into the other cell. This is called a rolling circle because it sort of unrolls here and rolls up into the other cell. Now you can interrupt. This is called conjugation. And if you know what that word means, you might understand why this is called conjugation. If not, we'll get to it in chapter 12. This is called conjugation. And if you interrupt conjugation at specific time intervals and then look at how much of your chromosome has gotten in there, you can see the relative times at which pieces of the chromosome entered the cell. So this map shows that when they did this conjugation map, this gene, THRA, entered first, LU entered second, Pro entered third, and these numbers in the middle are actually minutes. This is how long they let it go and when they found these genes in the other cell. So the map on E. coli is actually a time map, how long it takes for that gene to get into the other bacterial cell based on this rolling circle model. So let's say that we have an E. coli chromosome and we have some genes on it and we want to map where they are. We let this chromosome go through conjugation and infect another one, and we stop it at different times. And let's say that we get an order of D, E, F, G, H, I, A, B, C. Now, all this means is that around the circle, we have these genes, and they occur in this order. If the conjugation were to start here, we would get H, I, A, B, C, D, E, F. And so you just sort of get your order based on when they entered. Because there is no sort of same origin every time, you might get a different start point, but you should always get the same sequence. All right, going back to eukaryotes, we looked at genetic mapping or relative mapping, and this was based on recombination frequencies. But you can also make physical maps or absolute maps, which put genes at very specific base pair numbers. So you do this based on DNA sequencing. You get down to the base pair number at which each of these is located. So this gene here, BLI3, is located at base pair 1000, FOG1 at 3000, DPY5 at 5000. And you actually get very physical absolute distances. There's a specific number of base pairs in between these genes. Now this is compared to the genetic map that we were making, the relative map based on recombination frequencies. In this map, you get the same order, but you get different distances in between. And that's because recombination is more likely to occur at some regions over other regions. And it doesn't have anything to do with how far apart the genes are. It has to do with how likely a recombination is going to occur in any specific region. So some regions are affected by different recombination rates. Now you're going to get the same order, but you are not necessarily going to get the same absolute distance because you're measuring two different things. That's not to say that one is better than the other. That's not at all true. They are used for different reasons. All right, and finally, we can also make physical maps of the entire chromosome. So this on the side is a karyotype. We've seen that before. And karyotypes are used to make cytological maps. So a cytological map is just the dyed chromosome map. And from that, you can infer different regions, and you're going to use this to map physical characteristics of a chromosome. So you might use a cytological map to look for a damaged chromosome, or look for a chromosome with a piece missing, something like that. And so this isn't going to give you base level specificity or even gene orders, because this is a physical map. This is pulling way out, but it is going to give you important information about the physical characteristics of the chromosome.